So what I want to talk about today, this is kind of a fairly large generic topic, and so I'll just say up front that I'm focusing on postganglionic sympathetic nerves, and this is the least accurate, most oversimplified version of nerves in a heart that you'll see at this conference. I'm pretty confident of that assertion. So the only point that I want to make here with this picture is just to remind all of us that there are many different things that are going on. And what I'm going to talk about is myocardial infarction, and particularly most of all the data I'm going to show you today was done in mouse, and we're doing ischemia and reperfusion. So we're typically using the LAD, compressing it for, say, 40 minutes, and then allowing reperfusion. And when you do ischemia reperfusion in an animal where we can kind of look at these things in a different way than you can track in humans, what you see is that basically every kind of neuroplasticity that's been described anywhere in the nervous system is happening. And just even looking at the sympathetics, let alone everything else that's changing. So there are region-specific changes in neuropeptides and neurotransmitter content, and so that even in, say, the base of the heart, something can be going on that's quite different than farther down, closer to the apex. There's denervation, there's reinnervation, there are changes in neuronal excitability, so this could be the stellate or pick your ganglion of choice, and changes in gene expression here that are sometimes consistent with the changes in transmitters that you see in the heart, and sometimes seem to actually be conflicting because there's local things going on here that can override the changes in gene expression. So what I want to talk about today for most of the time is one very specific set of stuff we've been working on related to denervation and then reinnervation. And then I'm going to try, in fact, is there something here telling me the timing? So like flash me when I have like 5 minutes left, because I want to spend a few minutes talking about some newer data on neurotransmitter changes that's a very much still in process preliminary thing, and I'm really interested to get your ideas about some experiments that we should be doing on that. So many of you are familiar with these studies that have come out over the last several years. These are all human studies where they've done MIBG or C11 ephedrine imaging to image in humans sympathetic innervation. So what I should say, image functional transport through the norepinephrine transporter, okay, is basically, but that's a marker of kind of functional sympathetic innervation. And in these studies, then these patients received ICDs, and so what they did was then follow them later and ask, okay, who had arrhythmias and their ICDs fired and who didn't? And all of these studies, which were done in different parts of the world, found that sympathetic denervation in the heart. So we're talking about the loss of axons, right? This point's been made earlier. The cell bodies are in a different area, so we're talking about loss of sympathetic axons in the heart, that this seems to predict risk for arrhythmias. And one of the things that my lab found several years ago is that when we do ischemia reperfusion and then look at different times after that, the nerves grow back really well until they get to the area of the scar and then they stop. And this is very different than what happens in chronic ischemia. So here's a chronic coronary artery ligation, and the scar here, there's lots of Th positive sympathetic nerve fibers, there's hyperinnervation, which other people have seen. And the difference is these things called chondroitin sulfated proteoglycans, which for obvious reasons people call CSPGs. And so here we're staining these sections with a pan-CSPG antibody. So chondroitin sulfated proteoglycans are there's a whole family of core proteins that are modified with these glycosaminoglycan side chains that are sulfated on either a 6 or a 4 position. And these things have been best characterized as being indicative of reactive gliosis in the central nervous system. So spinal cord injury scar, traumatic brain injury scar, and it turns out that these CSPGs are a really important scar component that prevents nerve regeneration in the central nervous system. So it turns out that a reperfused scar in the heart is covered with these things, and not surprisingly, the TH-labeled sympathetic nerve fibers are excluded from the scar. So several years ago, after 20 years of work, the spinal cord injury people discovered PTP sigma, protein tyrosine phosphatase sigma, as a receptor for these CSPGs. So this is a transmembrane phosphatase. And so what we showed was that in animals that lack PTP sigma, and this is predominantly a neuronal protein, okay, that sympathetic nerves, now we do ischemia reperfusion, we get lots of reinnervation of the scar. And in fact, hyperinnervation, because there's a lot of nerve growth factor in the scar. And so here we're labeling with fibrinogen and tyrosine hydroxylase, and in the shams of these animals with the two genotypes, there's no difference. So innervation density, norepinephrine content, et cetera. And you can see here that it's not just the scar that's denervated, but there's an area around the scar that's also tends to be denervated. So we finished this a little while ago, and we were really interested to know what is this doing to arrhythmia susceptibility. So in that first set of animals, we had ECG telemetry implants. So here's just a couple samples of the scars which we could collect then after the experiment. You can see tyrosine hydroxylase intermingled with fibrinogen in the knockout, but not in the heterozygous animal. And when we injected isoproterenol into these animals, and this was a couple of weeks after ischemia reperfusion, we got significantly more PVCs in the infarcted animals, the control infarcted animals, but not in the knockouts. And we actually had this data a long time ago, and we weren't really sure what to make of it. We kind of put it aside and finished some other things first. And I will say, so the heart rate response to isoproterenol is identical. Scar size is identical in both of these genotypes. So then we followed up doing a series of experiments in collaboration with Crystal Ripplinger at UC Davis and Langguo Wan, who's a scientist in her lab. And we did, so the way we did this is we did the surgeries and then let the animals recover for a while, shipped them to Davis. We let them recover there for a while so that they were all happy. And so 3 weeks after reperfusion, ischemia reperfusion surgery, Crystal and Langguo did mapping experiments with these animals. So we're looking at simultaneously voltage and calcium. And in case you're an optical mapper and care about the details of the dyes, I've listed them here. And so the setup here is just Langendorf perfused heart filled with these dyes, and then collecting ECG information for the whole experiment, and then periodically stimulating the heart with lasers and capturing high-speed movies. So when we did this, so this is the basic setup. So there's a stimulating electrode up here at the base. All the pictures and maps I'm going to show you are in this orientation. So left ventricle on this side, base, apex, and in the hearts that have an infarct, the infarct is generally going to be in this area. And this is just some sample traces of the optical action potential. So these are mouse ventricular myocytes, so they have a more triangular action potential, obviously, than a human or larger animal. Now, mean action potential duration was identical across all the groups. But when we looked at interquartile range, which is a measure of dispersion of APD, not surprisingly, we saw that in our control infarct animals, there's an increase in APD dispersion. But interestingly, in the knockouts, where we've got reinnervation of the infarct and border zone, APD dispersion was identical to sham. Similarly, getting reinnervation, you would expect would prevent beta receptor supersensitivity, since that's caused by denervation. And that's exactly what we see. So here we're looking again at APD, and if we look at an individual spot here, map here before and after isoproterenol, where we're adding that to the bath, you can see there's a much bigger shortening of action potential duration in our animals with a denervated infarct versus with the reinnervation, and just quantifying that across 5 or 6 hearts of each group here. Now, something that was quite surprising to us was the effect on calcium handling. So here we're doing just a pacing protocol, pacing at kind of a regular kind of heart rate, which in a mouse is like 450 beats a minute. And so in the shams, you can see very nice calcium signal. It's very regular. And this trace is, again, just showing what we're seeing at the spot identified by this asterisk. And in our infarcted hearts, in all the infarcted hearts, we saw at this same pacing rate calcium alternans. When we did these experiments in the knockout infarcted hearts, which have the same size of a scar, and we're pacing at the same rate here, we don't see any calcium alternans. And that's quantified here with spectral imaging across 5 hearts of each group. And we had one knockout that had just a teeny bit of alternans, but as a group, they were not any different than the sham. And further looking at the calcium imaging, now we're looking at voltage as well as calcium together. Under baseline conditions, we're stimulating these hearts. The upstroke and voltage drives then the increase in calcium, and that is the same in both sets of animals. But in our control animals with denervated infarcts, when we added isoproterenol, so that we're activating those denervated beta receptors, we see calcium start to rise before the pacing stimulus. And in other data that I'm not going to show just in the interest of time, this leak in calcium does correlate with PVCs that start at the site of the infarct when we capture these during our mapping protocol. And so this just is quantifying that calcium transient that goes up, completely blocked by propranolol. And I should say, when we add tyramine to the bath, which is causing release of norepinephrine from the nerves that are in the heart, we don't see this. So we only see this kind of thing when we're adding isoproterenol, which is now accessing those denervated receptors. So the other thing we did with these ex vivo experiments is look at arrhythmias, and so just counting events here. But we also looked at arrhythmia severity. I have to say, this is one of those things that seemed obvious after reviewers pointed out to us, like, you should do this. We said, yeah, we should. Wow, good idea. So in the knockout hearts with reinnervation, basically the only thing we ever saw was a one-off PVC, which we gave individual PVCs, got a score of 1 here, and then 2 here is bigeminy or salvos of PVCs, and then 3 ventricular tachycardia. Now 4 would be ventricular fibrillation, which we never saw in the mouse hearts. And so what you can see is, and not surprisingly, when you're counting individual events, and you've got some hearts and ventricular tachycardia, the number of individual events happening in these head hearts that were denervated was way higher than everything else. And again, in both genotypes, all those things were blocked by propranolol. Okay, so what we'd really like to do is be able to fix these nerves in a way that doesn't require knocking out a gene, because obviously, I mean, that's not going to be very relevant in humans. And also, even though PDP sigma is not expressed in cardiac myocytes, it's just, one is just always a little concerned with knockouts that there might be some off-target effect that you don't know about. So over the last several years, Jerry Silver and Brad Lang, who are in neuroscience at Case Western Reserve, have been working on developing therapeutics for spinal cord injury. And Jerry is actually, I actually did my postdoc many years ago in that department. And Jerry's been one of the people involved in identifying CSPGs in traumatic brain injury and spinal cord injury. He was involved in identification of PDP sigma as a receptor. And so when we got into this field, we've been talking with him over the years. And so they contacted us some time ago and said, we've got this peptide. It works in cells really well, seems to sort of work in animals. We want to send it to you. Will you test it in your MI model? And so this is something they call ISP, or intracellular sigma peptide. So here's a little cartoon of PTP sigma. There's two theoretical catalytic domains. This is actually the active phosphatase domain. It's also a site for protein-protein interactions. So PDP sigma can dimerize with itself. It can dimerize with related receptors. And it also interacts directly with some of its substrates. And so this D1 catalytic domain is very important for a lot of those things. So what we did was implant our telemetry CG implants, make sure the leads are working, do ischemia reperfusion surgery. We waited 3 days because during this first phase, axons are just degenerating. And so we waited till that was over, and that's why and how that works is a whole nother topic. And then we started injecting ISP, just IP injections every day for 10 days. And then 14 days after reperfusion, we challenged the animals with isoproterenol. So first of all, here's a vehicle-injected animal, fibrinogen-labeled scar, very few tyrosine hydroxylase positive nerve fibers. Here's an ISP-injected animal, and we've got full reinnervation of the scar, quantified here across a bunch of sections from a bunch of animals. And we also see norepinephrine content is restored to normal in these infarcts. So when we challenged these animals with isoproterenol and counted PVCs, again, we got basically the same kind of response we see in a sham animal. And we have not injected anything into these animals until 3 days after the injury. So the infarct size is identical. And so for this part of things, chondroitin sulfated proteoglycans cover the cardiac scar after ischemia reperfusion, but not ischemia. And if you remove or inhibit PDP sigma, you can get reinnervation of the infarct. And let me just stop and say, we have looked at sympathetic nerves so far, but every intervention and all of these players also impact sensory nerves as well. In fact, that's the classic model for CSPGs preventing axon outgrowth. So it's entirely possible that we're also rescuing kind of reinnervation of that part of the heart by sensory afferents. And when we have reinnervation of the infarct, that normalizes calcium handling in electrophysiology, and it makes the hearts resistant to isoproterenol-induced arrhythmias. Now, we have more recently been doing some work with a colleague who's a chemical biologist who's very interested in neuroscience, and been testing some small molecules that he's developed based on this natural product, which was reported to interact with this receptor LAR, which is actually very similar to PDP sigma. And just very briefly, we've got two compounds now that seem to work really nicely. So for these things, we're just growing neurons in a dish. We have an automated system now. There's a new toy in our neuroscience imaging core we've been using, where basically it will take a bunch of pictures over a period of time, and you can kind of do a time lapse and watch processes grow in the cells. And so the software basically finds the cells and the processes, and we can calculate growth rates. So this is like one well in a 96-well plate. We can treat these neurons, and we can do dose response curves and things with these drugs. So in this case, the amount of CSPGs that we used to coat the plates decreased growth by about 50%. And this compound called HJ01, based on the initials of the postdoc who made it, at 100 nanomolar concentration restores axon growth back to control levels. And then this other compound at 30 nanomolars restores growth back to control levels. And if we add 2 more carbons on the end of this, it's completely dead. It doesn't do anything. So we have these 2 compounds. They both can fully rescue sympathetic growth at nanomolar concentrations. We know from data that I didn't show you that they do bind PDP sigma. What we're trying to wrap up is whether or not they're completely specific for PDP sigma, because they are also targeting that catalytic domain. So there's a reactive cysteine in that phosphatase domain, but phosphatase domains and kinase domains are often very similar. They all bind ATP, they all do, they do similar kinds of things. So we're very close, but trying to make sure that this is really specific and not cross-reacting with some other family members. And also, we very much want to know if they work in vivo. And we're working on trying to get money to do that, and I'm open to any ideas you might have for that. Now, the last thing that I want to briefly talk about, 5 minutes? Okay, perfect, is some stuff that's very much in process, looking at norepinephrine and acetylcholine changes in the heart. And we've also looked at some changes in gene expression. So I think many of you in this audience will be very familiar with this paper that came out several years ago now from the Fukuda lab, saying that sympathetic neurons basically start making norepinephrine, or start making acetylcholine instead of norepinephrine in heart failure, and that's caused by activation of GP130. This was fascinating to me, probably for a completely different reason than it might be fascinating to the rest of you. I spent my postdoctoral years with Story Landis working on sympathetic nerves innervating sweat glands, which become cholinergic. And in fact, part of what my work was back at that time was showing that GP130 cytokine signaling in the neurons was that transdifferentiation process. And it was later that I realized that the things I thought of as developmental differentiation factors, everybody else in the world thought of as inflammatory cytokines. And so that's when I switched more to the injury stuff. But this paper was really interesting, and we actually had mice whose sympathetic neurons lacked the GP130 cytokine receptor, which we'd been using for some other things. And so I went to the freezer, and I actually generated this data myself. I have to point that as probably the last data I've generated. Now I go in the lab, and they just hide, tell me to go away. But in the wild-type animals, so here we're looking at mRNA in the stellate ganglion 7 days after ischemia reperfusion. So at 3 days, nothing has changed. At 7 days, here's choline-acetyltransferase, the synthetic enzyme for acetylcholine, the high-affinity choline transporter, which is the rate-limiting step in acetylcholine synthesis. We've also looked at the vesicular acetylcholine transporter and a transcription factor that turns these genes on, and all of those are increased in the heart after ischemia, or in the stellate after ischemia reperfusion, and not in the GP130 knockouts. And more recently, we've gone to later time points. And here again, just showing choline acetyltransferase and the transporter. These are significantly elevated in the stellate, and we've been measuring acetylcholine content in the heart after ischemia reperfusion. And so we see that in the infarct itself, it's very stable, and these dissections are a little bit quick and dirty because acetylcholine is very labile, but we try under just a dissecting microscope and keeping the tissue as cold as possible to basically eyeball the scar and the area around the scar. And we do see significant increases in acetylcholine in the peri-infarct ventricle. Now, is this in sympathetic nerves? That we don't know yet. We have measured norepinephrine content. Now, we've developed a method to measure norepinephrine and acetylcholine from the same tissue. And norepinephrine content is low in the infarct because it's mostly denervated, but it's completely normal in this peri-infarct zone. So these norepinephrine goes down really early after MI, but now in this tissue, there's a normal amount of norepinephrine, but there's an elevated amount of acetylcholine. Now, still, the total amount of norepinephrine, much higher than the amount of acetylcholine. But the ratio seems to be different. And so we did this one thing with Crystal Ripplinger. So I think some of you are aware of some stuff that they published in Circ Research a couple of years ago where they injected norepinephrine and paced the hearts and found that norepinephrine increased the number of PVCs and reentrant arrhythmias and things. So basically, we just asked, if you do that experiment and now add acetylcholine, so you're injecting both in the same spot, what happens? And so far, it turns out not much. So it doesn't change the number of PVCs per injection, doesn't change the ratio of injections causing a PVC. They've tried injecting in kind of different parts of the heart to see if that made a difference. So far, none of this makes a difference, but this is just one like very superficial thing. So we're very interested to get ideas on what kinds of things we should be doing to try to get at if sympathetic nerves are making acetylcholine, you know, how can we unearth what's going on there? And so there is acetylcholine. We don't know yet for sure that it's from the sympathetics or what it's doing to arrhythmia susceptibility. Most of what I talked about today was done by Ryan Gardner as part of his PhD work. The mapping was done with Crystal Langlois, and Mike and the folks at CASE gave us reagents. So thanks.